باين ولا ايه؟ Okay, everyone. Okay, let's finish this lesson. So where were we? Yes, you can all hear me? We were talking about codominance. Codominance is another example of inheritance in which you will inherit two dominant alleles and both of them are going to appear on your phenotype. And the example in your syllabus is the blood groups. Now, you have blood group A, blood group B, and blood group O. A is dominant, and B is dominant, and O is recessive, in case you forgot. If you inherit two alleles for A, your blood type will be A. If you inherit two alleles for B, your blood type will be B. And if you inherit one allele A and one allele B, your blood type will be AB. And this is an example of codominance. Don't forget that the blood group O is recessive. So if you inherit allele A from one parent and allele O from another parent, your blood type will be what? If your blood type is A and O, you inherited one allele that's A from one parent and another allele that is O. What will be your final blood type?
A.
A, because A is dominant and O is recessive. And blood groups, yes, you can write it only by letters, okay? But more professionally, we use the gene I. I is for blood type, and the allele is usually written above the allele or the gene. Okay, so this is the gene and this is the allele. Okay, so codominance is the typical example, blood groups. And also there is another example that we see in exams in which you have a red flower. I think it's going to come next. No, we did talk about color blindness. Where is the red flower? The red flower, when it crosses a white flower and then you have a final pink flower, it is called intermediate dominance or also codominance. And we answered questions in the classified or in exams, and the answer that is given in the mark scene that it is codominant because both of them affected the phenotype. The phenotype. The flower is not white and it is not red. It is something in between. It's pink. In some books, they call it or describe it as intermediate dominance or just simply codominance. Okay, we did talk about color blindness, and we know now it's a sex-linked disease. It's recessive. Sex-linked means it's found on one of the sex chromosomes, usually the X. Sex-linked means it is a disease that is more common in one gender than the other, usually males. Okay, we said all this. Now, sometimes they ask you for a genetic diagram, but they're usually nice and they will give you guidance. Like they will tell you parent phenotype, and the phenotype is usually given. And then they continue and you have to fill it in. The parent genotypes, and then the gamete genotype, and then the offspring genotype, and then the offspring phenotype. So you have to follow the guide. Parents genotypes, this is the genotype here, the gene in terms of the alleles present in the gene. The gamete genotype here, the gamete is haploid. So the gene will be split. This is XY. This is a male with a allele on the X. This is going to be split to give you the gamete. This is the X and this is the Y. And this is the X and this is the X because this is the female. And then the offspring genotype. In here, in that step, you can do a Punnett square, or you can simply join the alleles together like this. This allele will join with this one to give me this gene, or this allele is going to join with this one to give me the second, and so on. Okay, but it's easier to do a genotype in this, a Punnett square in this step. And then the offspring phenotypes, I think it is very easy. This is a female that is either affected or not affected, and so on. Okay, we did this example of the homozygous dominant and homozygous recessive, like a test cross. In this combination, you always have all the children will be heterozygous. 100% will show the dominant allele. In this combination, heterozygous and heterozygous, the phenotype will be 3 dominant, 1 recessive. Let's do it. Here is a Punnett square, and you have a heterozygous parent and another heterozygous parent. These children, this child will be homozygous dominant, this will be heterozygous, this will be heterozygous, and only the last one will be homozygous recessive. So 3 showed the dominant allele, and only 1 showed the dominant phenotype, the recessive phenotype. So the ratio is 3:1. Sometimes the questions are tricky. They tell you that 2 heterozygous parents were crossed and they gave me offspring. And 2 from the offspring were crossed, or 1 from the offspring was crossed with another offspring. When you have this question, okay, you have to first cross the parents and then know the genotype of the offspring and then use one of the offspring and do the second crossing. So please read the questions carefully. Okay, we're actually done. Anyone has any questions in this lesson? This lesson must be understood, and you have to really understand the difference between mitosis and meiosis. Anyone has any questions? Hello? Anyone has any questions for the mock exam?
دكتور
Yes.
هو ممكن حضرتك بس تريفايز your 10 notes excretion.
Okay, I will. I'm going to go directly to the, we explained it in class, but obviously you were absent, or I don't know, where were you? Okay, we will go to the nephron, straight to the nephron. The kidney is the organ that here it filters the body, it filters the blood, and it contains many of those structures. These structures are not cells, they're called nephrons. This part of the nephron is found in the outer part of the kidney called the cortex, and this part of the nephron is found in the medulla. Now, this part of the nephron here, it looks like a cup, and we have capillaries here in the center. These capillaries are called the glomerulus. Okay, and we have an artery going into the glomerulus and an artery going out of the glomerulus. These arteries are not the same width, and the glomerulus here is under high, who reminds me? The glomerulus is under high what?
Pressure.
Pressure, okay? And because of this high pressure, so you have blood capillaries here in the nephron, in the Bowman's capsule, that are under high pressure. And because of this high pressure, only the small soluble molecules will be filtered out. And this is the first step in the making of urine, known as the ultrafiltration. What are the small soluble molecules? The urea, glucose, salts, and the water, the guess what molecules, and the amino acids. Together, they will make the filtrate. Who is asking? Hello?
أنا
Are you with me or you're still confused?
لا still confused.
Okay, I have to explain this lesson in a different online altogether. I cannot explain 2 in one day. So I'm going to give you another revision session on the night of the exam, on Sunday, which is actually tomorrow. Tomorrow, I'll explain excretion in detail from the beginning to the end. Okay?
Okay.
Were you absent when we were explaining this lesson? Because we explained this lesson over a week. Where were you? Excuse
في
me?
class. كنت في class.
Okay, fine. I'm going to explain it tomorrow.
Okay, thank
Fine.
you.
See you tomorrow. Bye-bye.